Hey all, welcome to Sharetreck and welcome back to our series on synthetic biology. This is the second episode of the mini series on synthetic biology basics for investors. Synthetic biology market is a growing market and it's quickly growing with prospects of great investment opportunities. Of course, at the start of an industry, there's going to be high level of risk because information is not generally available and the investor is not familiar with the technology of the target investment, and therefore an investor needs to understand a little bit of synthetic biology before considering investing in this field. At Sharetreck, I'm working on putting together basic information that an investor would need to have in order to reduce the risk of investing in the emerging field of synthetic biology companies. There are quite a few companies that are quoted in the market, and we want to make sure that while investing, we do not burn our fingers. I have many more upcoming episodes in this series to provide a good understanding of synthetic biology from an investor perspective. It's not like we are going to dig deep into this technology, but just enough so that we can understand what's happening. So if you are interested, please press the subscribe and notify button so that you don't have to miss any of these episodes. So with that said, let's get started. Today we'll be discussing the fundamental building block of synthetic biology, that is DNA. As you may already know, DNA is the molecule that carries the genetic information in living organisms. In synthetic biology, DNA is used as a tool to create new biological systems. One of the key techniques used in synthetic biology is genetic engineering. This involves manipulating DNA in order to add or remove specific genetic sequences. By doing so, researchers can create new genetic combinations that can be used to create novel biological systems. An important tool in synthetic biology is the gene circuit. A gene circuit is a set of genes that are designed to work together to perform a specific task. Gene circuits are networks of genes that interact with each other and with their environment in order to regulate cellular processes such as metabolism, development, and response to stimuli. They can be thought of as a set of logical operations that allow cells to make decisions based on external cues. There are many different types of gene circuits, but they can generally be categorized into two main categories: regulatory circuits and signaling circuits. Regulatory circuits control the expression of genes by turning them on or off in response to specific signals. For example, the LAC operon in E. coli is a classic example of a regulatory circuit. The LAC operon contains 3 genes that encode proteins involved in the metabolism of lactose. When lactose is present in the environment, it binds to a regulatory protein called the lac repressor, which then releases its grip on the operator region of the operon. This allows RNA polymerase to transcribe the genes and produce the proteins needed to metabolize lactose. Now, this is a scientific explanation. For us lay people, I'll give a much simpler example. The way yogurt is set is first you boil milk and reduce its temperature to a room temperature level, and then you put an inoculum of yogurt into it, a small bit of yogurt into it, and then the bacteria starts working on it, and when the bacteria reaches a particular threshold, it secretes certain enzymes which converts the milk into yogurt. So that would be a layman's example I would give for the scientific explanation that I read out a few minutes ago. Signaling circuits, on the other hand, allow cells to communicate with each other and respond to changes in the environment. One example of a signaling circuit is the quorum sensing system found in many bacteria. Quorum sensing is a process by which bacteria can sense the density of their population and adjust their behavior accordingly. Bacteria release small signaling molecules called autoinducers into their environment. As the population density of the bacteria increases, the concentration of autoinducer also increases. Once a particular threshold concentration of autoinducers is reached, the bacteria will activate genes that allow them to coordinate their behavior, such as forming biofilms or producing virulence factors. And I would say that yogurt setting is also one of those examples of having reached a threshold and then performing a particular function. In summary, gene circuits are powerful tools that allow cells to sense and respond to their environment in a coordinated and regulated way. Understanding the logic of gene circuits is critical for developing new therapies, engineering biological systems, and improving our understanding of fundamental biological processes. Now that we have an overall understanding of gene circuits, it's interesting to know that they can be synthetically engineered. In order to build gene circuits, synthetic biologists use a variety of techniques to manipulate DNA and create novel genetic systems. Cloning is a common technique used to copy and amplify DNA sequences, which can then be inserted into host organisms to modify their genetic makeup. Gene synthesis involves the assembly of artificial DNA sequences that can be used to create entirely new genes or modify existing ones. Genome editing, which has become increasingly popular in recent years, involves the precise modification of DNA sequences within an organism's genome, allowing for the targeted addition, deletion, or modification of genes. These techniques are often combined with computational modeling and analysis to design and optimize gene circuits for specific applications, such as biosensors, drug delivery systems, and biomanufacturing processes. Together, these tools and techniques are transforming our ability to engineer biological systems and create novel solutions to complex challenges in healthcare, agriculture, and beyond. These techniques allow researchers to manipulate DNA at a very precise level, making it possible to create new biological systems with specific functions. While DNA is the fundamental building block of synthetic biology, it's not the only tool that synthetic biologists use. In fact, many researchers are now exploring the use of RNA and proteins in synthetic biology as well. With that, my friends, we have covered the basics of DNA and genetic engineering in synthetic biology. We could go deeper, but we don't want to, because as investors, I don't think we need more than this at this point of time. But if one of our target companies comes up with some press release with more information, and if we need to dig deeper, I'll definitely dig deeper into it. But right now, this is all we need to know. In our next episode, we'll be discussing the design principles that are used to create new biological systems. If you're a genomics investor, you would like to view these videos and find them very helpful, and you can revisit them anytime to brush up on the concepts by visiting our homepage in YouTube and searching for this particular video. I'm also going to make a separate playlist for synthetic biology, so you'll find it in the playlist section as well. So please make sure you subscribe and press the notify button so that you do not miss the next episode. And also, if you are not yet subscribed, why not subscribe? It's absolutely free. Subscribe and press the notify button, and you'll get to know as soon as I drop any new videos, and you can choose to watch it at a time of your choice. Thank you for joining me today, and I look forward to seeing you next time. Bye for now.